Good afternoon, everybody. It's a pleasure to be here with you all for today. My name is Christine Beveridge, and we will make a start in a moment. Sorry about that. I'm sorry about the slides flicking back and forth there. It wasn't a great start. I'm the Director of the Centre for Plant Success in Nature and Agriculture here at UQ, and it's my great pleasure to be here today. To begin, I'd like to acknowledge the traditional owners and their custodianship of the lands on which we meet today. On behalf of the traditional owners, I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. There are some housekeeping matters that I need to mention. This seminar is scheduled from 12 till 1 p.m. And at the conclusion of the seminar, we'll hold a question and answer session. If you have questions that you would like answered, please use the Q&A button at the bottom of your screen. Please do not use the chat function because we will not monitor that. We will use the Q&A questions and answers. And we will address those at the end of the seminar. So it's my great pleasure to introduce Jim Hanan. Associate Professor Jim Hanan's major research theme is in the development of mathematical computer graphics and simulation approaches and techniques that facilitate the study of genetics, physiology, morphogenesis and ecology at the scale of cells, individual plants and insects and their components. Jim started at the University of Queensland in 1992 and ever since he's had research focused positions, which I think is an incredibly impressive ability to have sustained that throughout his whole career here up until today. He started with the CIC for Tropical Pest Management, and then he was Principal Research Fellow in the Centre for Plant Architecture Informatics, which was a joint centre between CSIRO Entomology and UQ. And then he's gone through a range of other centres and now in coffee. And he's published incredibly diversely, actually, in plant science, in computer science, interdisciplinary applications, agricultural science, ecology, agronomy, mathematics, horticulture, entomology, and so on. So you can see he's incredibly diverse in his background and has an incredible 143 co-authors in his experience, and he's been a mentor to many of those people, and he's described very frequently as a team player, a highly collaborative individual, as evidenced by the things I've just said. And I think that's quite challenging thing to have achieved, and this to be really truly multidisciplinary. So I'm really looking forward to hearing this presentation from Jim. So over to you, Jim.
I'll be there in a second, as soon as my cursor comes back. Oh, there it is. Okay, so this is where I come from. Oh, thanks, Christine. And I should also add my acknowledgement of country. Start my video. Okay, good idea. Video is started. Okay, good. We're back. So this is a place I came from all those years ago. Saskatchewan. It's very flat, and in the winter that would be not sunflower fields, that would be snow. So I've been very happy here in Queensland. But this is a computer-generated image, and it's where I come from, is computer graphics as well. So what got me involved in computer graphics was getting involved with what was called fractals. Very intriguing, natural-looking shapes, and they had a lot of self-similarity. So this is called a Mandelbrot set, and it's iterated complex function, and the colors come from how far away the point is from the set. The black part is the set itself. But the interesting thing about these things is that when you zoom in, you can see the same sorts of detail at all sorts of scales. You could keep zooming into this. If you find one on Wikimedia, you can have a lot of fun with it. So another way of making fractals is this kind of construction we have here, where we have a straight line. Okay, maybe I'm gonna get the, you see my cursor there? There we go. Okay. We have each straight line, and it's going to get replaced in each step. With this straight line, it's going to get replaced by one with a little bump on it. That happens to each straight line in the image. And so then in the next step, the same thing happens. And this goes on to infinity, and then you have a fractal. If you stop here, it's not quite fractal yet, but you can also do nice things with it. With coloring and various sorts of things like that. So these are very intriguing images, but it also turns out that there's a lot of fractal in nature around us. So if you look at a branch of this tree, this one's not so good. It's maybe a bit of a, this one's better. That structure there is similar to the entire structure. So these kinds of images can be created and represent nature around us. Now, of course, the tree doesn't go to a limit and become really fractal, but it's got that fractal nature. But this kind of, being able to create this kind of image lets us go after images like this, which is called the Garden of El. And it's one of the earliest things that we did at the University of Regina in Saskatchewan. And where I was working with Professor Krushenkevich. We looked at that name and we affectionately called him Dr. P. I've worked with him ever since and still do. Sort of towards the end of my time there, we created this book called The Algorithmic Beauty of Plants, and coffee table kind of book was the idea, and it was went quite well for us. But again, it was about beauty of nature as represented by these things that could get towards fractals. But I have to go back a bit before that book to find Lindenmayer. Lindenmayer was a theoretical biologist who developed a formalism that described how living plants grow. And so he started off with blue-green algae, and he was looking through the microscope and seeing cells. So here we have a small cell, larger cell. They were divided into different kinds of cells, but the string of cells represented one of these anabaena. And so he developed this system that what he decided was you could represent these individual cells by different letters. So if you have A for the small cell, B for the grown version, D for the larger, darker one, and C for the smaller one. And then, starting from an axiom, A, being mathematical, of course, and having a set of production rules that that arrow is read as produces, A produces B, and you can see that in time step 1 to time step 2. And then B produces DA, so we go from B, DA. And if you follow this set of rules, you will find that the alphabet that describes that string comes out of the application of these rules. So this kind of formalism was developed from Chomsky grammars. He was reading the computer science literature of the day, which was very young, but he decided that rather than do things sequentially, you could do them in parallel. And that's what is the key defining thing of a Lindenmayer system, which became, because people were bored of saying systems of Lindenmayer or Lindenmayer-systems, they started calling it Lsystems. So how does this work with plants? Well, here's a plant, nice and neatly drawn. And here's the alphabet of symbols we're going to use. An I for an internode, an A for an apex, L for a leaf, no matter what size. And that will describe our structure. So we've got these connected modules described by the alphabet. Then we put them in a string so that the computer can handle them, of course, or that we can apply the string rewriting rules to create the next structure. So here we have the internode I, and then because we've got three things connected to that, we have a leaf, an internode, and an apex, and the ongoing internode. So they appear next to that I, and the way they're divided off into their branches is with these branching symbols, square brackets. So you can read that as start branch and end branch if you like. So then we have a set of rules, these production rules, where a predecessor becomes a successor. So here we have a visual sort of version of it. The apex produces a metamer, which is an internode, auxiliary bud, the ongoing bud, and the leaf. So then we have the rule, A produces I, start branch, leaf, end branch, so on. So then we have B matures into the apex, leaves grow, and internodes grow. So then if you apply these things from the axiom, the starting point, and you apply them step after step, you get a growing structure. So you can see that the way the computer model runs it. You can have it here. This is a program called CPFG that's running this. And we can have sliders. And here I'm just changing the angle from one stem leaf to the next. And you can see that you get a different type of plant out of the deal. And rotating it lets us look at the 3D nature of it more easily. So how do we get the structure? Well, we interpret the string using what's called turtle geometry. This is something that came from Logo, which is a language which I forgot to look up where it came from. And what they had was a big ball on the robot kind of device connected to the computer by an overhead cable. And it had a pen in it that could go up and down. And so when it got directions, if you see, you can start it off at the bottom here, facing upwards on the piece of paper. Then it gets an instruction, move forward for length of 2. So that's this move. Turn to the right, that's the minus sign. Then move forward for length of 1. Turn left for 45 degrees, and then move forward for length of 1. So we have rules like that describing each component. You don't usually think of them, the details of that, because you have a set of interpretation rules that help you along. So yeah, how do you get the information in there? Well, here we see a younger version of myself using a sonic digitizer to digitize a bean plant. It doesn't look like it is a bean plant, but anyway, let's call it a bean plant. And we have a program on the computer, running on the computer here, called Flora Digitizer, and it lets us capture the 3D coordinates of each of the points, and then using sound, where it's measured from these three microphones to a little spark plug that comes from here. And then those coordinates give us lengths, widths, and so on. Then we encode those in rules for the plant. And again, we can rotate it in 3D. Here's this idea of how it works. And we can run the more realistic model here. The surfaces there are what are called bicubic surfaces, and they come from computer-aided design. We haven't been into this. It's nothing I did that in my master's. The top part of this model is deterministic. It's always the same. And whereas the bottom part, the roots, are actually stochastic. So to simulate the movement through the ground, they get into rocks. They got to move. That's the stochastic nature of the rules there. You can also do things like changing colors. And in this case, we've got generalized cylinders that are being used to represent the leaves and petals. And we can see we can use a widget there to change our colors from whatever we like, get a different variety. So those are the kinds of tools that you have in the system. So we have to get a little bit more, this is the sonic digitizer microphone array. We had to get cotton plant in there, we had to get a little bit more, build this kind of rig to get us going. So then we've got the structure by measuring. Miriam is there measuring it. And then we had physiology captured by the Oscott cotton crop model by Brian Hearn. And when we combine those two things, here's the structural model. On the left here we have the ideal development of the plant, where if everything grew, that's what would happen. And on the right here, we see a couple of actual specimens. This is data representing where the flowering points are. So we model potential growth in the structural model, and then we allocate daily crop model output positions using heuristics derived from observation. So these are three different specimens from a particular Delta pine growth simulation that we did with Oscott. So here's our cotton plant, and here's the kind of model that we can get out of the deal. So here we have those bicubic surfaces with texture maps on them, more stuff from computer graphics to make things pretty. Doesn't make any difference to the actual physical structure, if you like. So the years move on. We start looking at trees. We need fancier digitizers. So this is a Polhemus magnetic digitizer. And Neil White had a crew that went out down to down the coast, or up the coast, and measured this. It actually took a couple of days using the same Floridage software, but that's the structure that you get out of it, with color-coded by year of growth. You can use the same thing with mango. This was Anahita Mazzani's work with Ian Bally in Mariba. And so here we've got a particular heat leaf, set of leaves, a flush. And so these are the lengths of each leaf in the flush. So once we've got those into our model, we can have an animation that looks at whatever, in this case, heat and KP, Kensington Pride. So if we put those into a tree structure, we can have the flushes occurring where they occur in the tree. So what we have then is a structure of the tree. We can see here on the top left, top right, we have the flushing material, and on the bottom we have the side and top view of the complete canopy. So that's straight structure. So, but what else we can do? We can look at how information flow controls this development. So we can have endogenous factors like hormone signaling, transporter resources, biomechanical processes, or exogenous factors like environment management practices and things like that. Let's have a look at the endogenous factors. So to do that, we need to go to a bit more complex type of system, which Lindenmayer developed at the same time period. Here, the rewriting rules now depend on context, not just the symbol itself, not just the predecessor, but what's to its left. So this is left context, left of predecessor produces a successor. So when this particular context matches, this production rule will fire. So if we have an internode, we have our axiom starting structure with a signal at the bottom of the plant with 3 internodes. And our rule that says an S to the left produces an I with an S to its right. So then in step 1, we'll move that signal up, 2 and 3 till the signal goes through the structure. And so then you could imagine that a leaf or apex could pay attention to that signal and do something when the signal reaches it. And in the same way, we can have right context, where the predecessor, if the right context matches, in this case the S on the right, it'll move down through the structure in the same way. So here we're going to see a plant growing, and then as it grows, it's producing buds to the left and right. And then it will have a signal that comes up from the bottom. And when it reaches the top of the apex, it will cause flowering. And flowering will then release a signal that releases the buds below it, and they'll grow out and do their own similar process. So one set of rules, signal going up. When it reaches the top, it flowers, signal comes down that releases side branches. Oops. I'm going to go back. Here it's growing up. Now the signal's coming. It catches the top, producing a flower and a fruit eventually. And the same process happens in the side branches. So this is a weed called Lsystems moralis. And so there's a bit more realistic structure on it. And here we can see the part where it's doing the flowering. And we get a characteristic pattern where the oldest part is in the actual youngest part of the plant, the top. We have the seeds and fruit up there. And then in the middle, here we have flowering structures that are in the middle of the plant. So we can use the same kind of systems to look at how insects are moving around. So if we have that signal, the internode says, oh, there's an insect over there. Let's pull it on. In this case, we have 2 that are going after their plant. They like to eat the apices of the plant, and of course, causing damage. It won't grow any further once the apex is taken away. So what we'll see here is a difference in timing of damage, and it's going to cause difference in structure. On the left, it goes earlier. The right is delayed a bit. So you can see that that structure is a bit bigger. These guys are really, really slow. They're only moving as fast as the plant is growing. But anyways, it demonstrates the idea. So we can think about things in the cell just as well. Here, the yellow represents a piece of the cell wall. You can imagine the cell off to the right of that image. And things to the left are on outside, things to the right are on the inside. So we have a receptor that goes through the cell wall membrane, and where the Y is, it captures that blue signal and passes something through the cell wall, which through phosphorylation, adjusts the structure. Now the little fishhook guy up on the right is trying to catch the right kind of signal. So when it matches, it'll take signal away with it. And the guy down on the bottom is going to sort of steal signal when it can. So here we got our orange ball coming across, making the signal, and you can see it's gradually bending the molecule over. And, oh, not quite the right shape yet. Not enough signal, and it'll take away that signal to the nucleus, for instance. And here now we've got less signal, so the phosphorylation is being stolen, and so the signal doesn't happen anymore. So we can also model biomechanical processes. So this is a ray trace image. It looks very, very pretty, by Mick Cieslak, who is a PhD student of mine. And in this one, you can see right in the middle there's a blue bud and a red bud. And they're going to, when they grow, they're going to push into each other. So one of the petals. So in a normal computer graphic, you'd end up with those things crossing over and intersecting each other, which doesn't look very good. And certainly if you want to have the proper light interception, you need to have the leaves in the right places. So this is a rather advanced system that is going to, you can see now where the red and the blue petal are running into each other and they're not penetrating. So this is quite a bit of computer power to make something like this go. So that's sort of an example of the endogenous factors we can look at. And here, let's have a look at some of the exogenous, external factors in the environment, management practices, and so on. So now what we have to do is we have to get information from the outside to the inside of the plant for it to respond to. So we need to introduce some queries to the environment to get information back. So these queries will go out with their location and some information about the size of the leaf, for instance. And external program that represents the environment will calculate the external influence that goes back into the X. So here's an example of one of those where it's an overhead view of a patch of Earth where we get different light intensities. Bottom left, quite bright, less so in the top right, and right in the middle, it's quite dark, so nothing grows there. This is Clover. Oh, and I should have said, let's start that over again if we can. Oh, well. In the light, it makes very narrow branching angles with shorter internodes. So it populates quite heavily. And in the darker, well, it doesn't grow at all. Or it makes narrower angles here and longer internodes to find the light in the system. So we can have a look. So these die out eventually, and so you get a characteristic patterns of where you find the clover in your lawn. Here's another overhead view, this time of a piece of forest. We've got different colored circles representing trees. So the blue is a cone, the green is a sphere, and the white is an inverted cone. The red ones are things that have been outcompeted, and so they've died. So the competition comes when trees intersect, and the vigor, which is determined based on age for different varieties, whichever has the highest vigor survives. Now the green ones actually don't die; they just go a bit dormant and wait. So it's quite common in rainforest plants. So what you can see here is different sizes in this graph on the right. So radius up to the high radius over here, and those are the numbers of things. So you can see here we've got spherical and cone-shaped trees. So these create characteristic age-related patterns in the forest, the patch of forest. We can look at plant insect interactions. In this case, we're going to have butterflies that come in. They, based on their, what do we say, fuel load, or the amount of how hungry they are, they'll fly to the flowers and refuel. And they tend to stay around those. And the poor bean plants that are closest to the flowers end up getting eggs laid on them. Eggs are laid when the insects get old enough. You can see a little 0 beside that guy. And you're not old enough yet to be laying eggs, so they're just flying around until they're 0 again. And come back down. Well, now they're laying those little red dots, which are their eggs. So if you were trying to determine the effect of these particular weeds in your system, you'd run a much larger field, much larger sample, and then have a look at the patterns that are created. I think it's a bit exaggerated example on my part, but I think you get the idea that there you can see the red dots and where they are distributed in the crop. So a big important environmental effect is light. So the plant simulator, in this case, generates the structure, passes out its inquiry to the light simulator, which then returns the light quantity or quality. So what we do is called path tracing of photons. So a light simulator called Quasi-MC, written by Mick Cieslak, takes the leaf geometry, puts it into an environment, passes the light through it, and then returns the light distribution. The photosynthetically active radiation, red-fired ratios, anything like that. So we stochastically sample photons. So for direct light, we're looking at light from the sun itself, or light in a glass house. And then you shoot lots and lots of rays, see where they intersect with the leaves, and then you have transmission with that little green part, and reflection, and then you have absorption, and that's the amount that will create photosynthate. You can have directional lights, or in which case you get more solid shadows, or you can have light pass sampled from a sky model, and you get softer shadows, and you can do things like integrate over the day. So here we have light distribution in the canopy. So the low is the red, high is green, and light is directly overhead. So interestingly, you've got, because the light is directly overhead, leaves that are hanging straight up and down don't get much light. And then if you do the same thing with a sky model, you'll see that most of the leaves get a bit more light. So we've, Jian Kao and the current PhD student, and Li Han, have been working on high performance computing to help us in these areas, because a lot of this takes a lot of time because you've got to shoot so many rays. So we have a high performance light simulator that, looking at these kind of between the serial thing that takes 13 minutes and Quasi-MC, which takes only about 4, and that's with the 4-core PCs. So if you get something with a few bit more, then you can go even get it even bigger efficiency improvement. So once you've got the light at each leaf, you can then do things like look at how much photosynthate is produced, how it's passed around in the structure, and therefore where the growth occurs and how much is allowed. So these are source sink models, and look at, in this case, we have a single pool carbon model that's looking at a avocado annual growth flush, if you like. You can see that we've got second and third order axes on here, where we have buds that are coming from the older wood, and then those that are coming from the new season's growth. And so these kind of models have been validated against some field data got from Bundaberg, Helen Hoffman. And Ming Wang is the guy who made the model. Okay, we have Inigo Azmendi here in town, who's also worked in a little slightly different kind of space. In this case, it's a macadamia tree, but looking at how autonomous units within the structure perform. So if you think about the carbon that's being produced by leaves, is it shared through the whole tree? If you look at this one A, that's where the structure is, the pool of carbon is for the whole structure. Whereas in this one, it's just for limbs, branches, or branchlets. I'm not going to talk about this anymore because I think Inigo is giving us a seminar next month where we can learn all about this kind of thing. One of the more detailed levels of carbon allocation modeling we do is called carbon transport resistance and allocation. The CTRAM, carbon tram. So this is consistent with the Wundt hypothesis at the scale of individual components, and we move things through the structure using this kind of physics. And this is work I've done with Alice Ellsnova in New Zealand and Mick Cieslak, who is here doing his PhD in this space. When you try doing these things, this is a bit of a model that is the same length in total, but with different breakdown, you can end up with different answers. So what we've done with ALA is look at having a more detailed model within the segment to get that time to be more correct and match no matter how many segments you have. Okay, well, these things are useful. So we've looked at Lsystems from structure and looking at internal signaling, including genetic regulation. You can have models of that as well. We've had plant-plant and within-plant competition. So we've had the carbon competition and the plant-plant competition where things were shading each other. And we've looked at insect-plant interactions. And for the future, things could be, oh, we got a virtual world. We could have robotic simulations in there and whatever. So an important thing, though, is to remember that what we're doing is computer experiments in silico. So we're taking some hypotheses, designing a model, running it on the computer, and analyzing what's the outputs, coming up with some kind of conclusions. Those feed back into new hypotheses for experimental design in the real world, in field experiments. And once we've got the results from those and some data analysis, we can decide if our model is doing things. We can't just look at the model and say, oh, this is what's going on in the plant. We need to have that feedback from the field experiments to make it work. And hopefully we have an upward spiral of increased understanding. So I'd like to acknowledge people here at the Plant Modeling Lab, Neil White, Inigo Azmendi, Lichi Han, some people from earlier, Michael Renton, Anahita Mazani, Ming Wang, and others. There's quite a few. DAF Queensland in our current work, John Wilkie, Ian Bally, Helen Hoffman. I've left a few off there as well. Collaborators, Dr. P, Mick Cieslak, and Pascal at University of Calgary, Alice Ellsnova at Plant and Food Research, who, like myself, is recently retired. Fred Boudon and Frederic Normand from INRA, and others at INRA as well, we've worked with in the past. Christine Beveridge in lab, Miran Zalitsky in lab, and here in the school, and others too numerous to remember. So I should say, though, that we've used this software, which partly comes from my PhD work, Vlab on Unix type systems for Mac, and LStudio on Windows environments, which are freely available. And there will soon be an open source distribution. So you can get in there if you're a programmer and try and change things to make them better. So thank you very much. And see if there's any questions. This quiet place here is a sort of like a picture we did early on, reminiscent of Monet, and that's very much all about what we were doing at the beginning. But we've used these Lsystems now to help us understand how the plants are growing and what effect it has that whether we have a weeping willow or a upstanding eucalyptus, and how it affects the physiology. So, thank you.
Yeah. Thanks very much, Jim. I really enjoyed that presentation, and I think this last image is just amazing how that looks like a painting, and yet it's a computer L-system-driven image. It's just amazing. So we'll open it up for questions now. There's a few coming through. The first question is, how accurate have computer models proven in describing field experiments? So, Jim, could you just comment on what sort of accuracy can you have in these sort of models in relation to the reality of field situations?
How accurate? Yeah, well, I guess they're, in some cases they can be very accurate if you measure a lot, and you can just represent exactly what you've measured. So it can be very accurate. But if you're looking at then a developmental model and thinking about how it interacts with the environment, well, there's lots of randomness involved. So you need to do large virtual experiments and compare them to good field data that is related. How well have we done? I think we've increased a lot of understanding of things by generating ideas that are then tested in the field, and yeah, I think we're accurate as we can be with the data that we have available to us. How's that?
That's fine. Jim, I just thought we might exit that full screen so we can a bit on full screen.
Sorry?
Yeah, just close. Oh, no, don't do that. I was just trying to figure out how to pin you so we could see your image a bit better, but that's okay. Yeah, I think one of the other questions was really about how well do you think the models can actually, you described the L-systems and the iterative, fractal type nature of those L-systems in describing the architecture of a plant.
Yeah.
In your experience, how well does that sort of, how well is that actually able to recapitulate an actual plant, given that you can throw in some stochastic properties in there as well? I mean, I think the images you presented are incredibly impressive, and they look very plant-like, right? So, yeah, one of the questions here is just how well does it compare to actual tree architecture?
Yes, as well as you want to measure.
Yeah, that's what I thought you were going to say. Measure, and I just, yeah, it's as good as the data you put into it, isn't it?
Yes. So the issue is, well, like with the trees right now, that we're doing measurement by hand of individual leaves and individual locations of leaves is quite time consuming. 3 days to create that tree architecture. So we're looking at more modern technology. Lsystems is actually looking at using LiDAR, which is big in architecture, and scanning from the back of a ute and seeing how well we can capture the kinds of information we need to get things at a orchard scale, if you like.
Mm-hmm.
For smaller things, you can measure them in great detail. And actually, you, Christine, have measured growth of buds and things in the lab at quite detailed levels. And if you can get enough of that kind of thing, and well, a good enough idea of why the things are changing, then you can put it into a model.
Mm. So, you know, for our centre that we're looking at trying to do some predictive kind of work. One of the questions that's come up is relating to, you know, how much can we use this sort of technology to bring many environmental signals and a multitude of things together. I mean, more than a human brain. Where do you see the future going with this sort of technology?
Yeah, well, of course, you're never going to have a complete model of the tree or a plant because it would take longer to run than the plant would to grow. So what you're doing is focusing in on particular issues or particular relationships by having a model. And just like any good experiment, you make things outside that not affected and see what the implications are of particular ideas that you have. And the model helps in developing the ideas, but then you have to go again and test it in the field. So I don't think we're ever going to have all the factors. And in fact, I would leave out some factors on purpose because I don't want to bother. If I'm doing carbon transport, maybe I don't want to have details of the root system, for instance, because I'm only interested in carbon transport from the leaf to the flowering bud to see how it's affecting flowering, for instance, so that the transport might be also of signals that are local at that scale. So I don't really want to have the whole system in there. So yeah, you're always going to be leaving something out.
Yeah. That sort of ties into this question about computer power limiting. So computer power, since over your career, computer power has sort of become greater and greater and greater over time. And I assume that's giving you greater capacity to do things. Do you still see that as a major limitation though, even in the longer term?
Well, because you always want to add something, don't you, and make it. So now we used to just do individual plants. Now we try to do whole orchards of plants. And if you want to have the right number of samples to get your statistical validity, you still need lots of samples. So those kinds of things move us along, I guess.
Yeah. And so relating to that, how much do you think industry will start to pick up on this sort of technology and using it for commercial use?
So I think definitely that they're already using the ideas that are being generated by models.
Yeah.
So whether they know it or not, their modeling is part of what they do. I don't think we're ever going to have a system that's going to say, oh, don't cut that branch, you should cut that branch over there for a particular instance of a tree. But it will give you an idea, here's what happens if you do this kind of cut versus that kind of cut. It should give you a bigger picture of what's going on. So again, it's about understanding, not about, it's not going to be an individual guide to, okay, cut this branch.
Yeah. I think you've also applied spray technology. So I think you had a model that allowed you to test how to spray for disease.
Yeah, we've got some spray. That's a very good point, actually, because Li Han is also running a project where looking at runoff and how spray is being captured in orchards. He's going up later this week to Mariba to look at the spray technology and see how that goes. So just like we have photons for light, we can have droplets of water or pesticide or whatever, herbicide, or flying through the air and intersecting with our canopies. So again, it's about ideas, and it's not going to give you the magic answer, but it's going to give you an idea which field work to do to find that answer.
Yeah. Not just the last question, I think, is you mentioned at the beginning about Anabaena, and you talked about insect-plant interactions. Is there much research being done, or using this sort of technology for that currently? Like, are people using this a lot for studying insects and relationships between plants and other organisms?
I wouldn't say it's a lot. There's been some work. Ming Wang did some work in that space, and we've done with Jürgen Zalitsky in his lab some work. But I don't think there's a large group of people working in that. Yeah, it's sort of an idea that hasn't had its time yet. Well, because we haven't been able to say, if you want to talk about insects moving through an orchard, you have to be able to model the orchard, so we're only just getting there.
Yeah, it's very early days even now. Look, Jim, I think we should probably wind up. I just want to thank you so much for that overview, really, of the research you've done over many years here at EQ. And I just want to remember too, a couple of the students that you've had that have really, I think so many people have benefited from your mentoring. I'm one of them. I think the centre that we've got funded, actually, there's this mathematical part of it is because of the mentoring in part that you've given me over the years and made it so much more accessible. And this idea of that diagram you had about the modelling and the testing in the field, I think that really is the future of how we should be thinking about things. And I'm thinking of Birgit Locke. She really wanted to come to your retirement yesterday party, but she was in lockdown. She's the deputy provost now, as you know, for TNL at the tribe, and I think that's an example of the sort of thing that you've been able to foster in people. And Michael Renton, teaching over in University of Western Australia. These people really have benefited enormously from your mentorship, and I just want, on behalf of you here and just today, just thank you so much for all that. It's been just amazing, and I hope that you will hang around.
That's the plan.
That's the plan, a little bit, and collaborate with us, because we really need your wisdom in these things. So yeah, thanks very much. And it's just a shame that we're not all here to sort of actually clap a cheer for you online with COVID. It's
thanks,
really
Christine.
frustrating. So I'll just finish off by saying that next week's seminar is titled Omics, Technologies and Vaccines, and will be presented by Professor Hanan from Coffee's Centre for Animal Sciences. And you can visit the Coffee Science Centre webpage to find information on that. I just want to thank everybody for attending, and there's a poll that you can enter to just give some feedback on this. And if you haven't already signed up for our seminar invitation list, you can do so at a link which is there. I don't know if that's the next slide, Jim, but coffee.uq.edu.au science seminars. So thanks everybody for attending, and thanks Jim again for just such a wonderful presentation. So beautiful images and so on. A great story. Thank you.
Thanks, Christine. Bye.
Bye-bye.